This is a production of Cornell University.
So today I'm going to be talking about a project that I've been working on for about the past year and a half. I'm just in the middle of it still, so don't have any end conclusions, but I hope you enjoy listening to my progress. So first, I'll start off by talking about the project that I'm working under. So the PhenoApps Project is a group of computer programmers and plant scientists who are working together directly with breeders to develop apps, smartphone apps for the phenotyping of plants. These apps will then be deployed through breeder networks, especially in developing countries. And one of the crops that the PhenoApps Project is especially interested in is cassava. So we talk a lot about cassava in this department, but thought I'd go over it again. Cassava is both an industrial and a food crop, and it's a staple for over 800 million people, especially in the tropics. Primary breeding targets of cassava are pest and disease resistance, yield and field performance, and dry matter content. Today, I'm going to be focusing mostly on dry matter content. So cassava root dry matter content is the proportion or the percent of the root that is not water. So after the water leaves the root, it's the percent that's left. So it's typically around 20 to 45%, and up to 90% of that can be starch. It directly affects the dry yield of the cassava roots and also the food quality or the mouthfeel, which then affects the consumer acceptance and the variety adoption rate of cassava. And in talking to breeders, I've found over and over that this trait is notoriously difficult, time-consuming, and laborious to measure. The two methods that are typically used are specific gravity and the oven method. The specific gravity method here on the left involves weighing the roots in air and in water and then plugging those values into an equation to get the dry matter content. The oven method requires transport from the field to a laboratory setting where the samples are dried in an oven after shredding, and this requires consistent electricity, which might not always be present in every given location. And so because of the challenges, the time put in and the labor put in, different breeding programs have been looking at a new technique to phenotype dry matter content a little quicker. And so this is near-infrared spectroscopy, or NIRS. So it's been shown to be highly predictive of dry matter in cassava roots, and because of this, it's being routinely used at CIAT in Colombia for the phenotyping of dry matter content in cassava. But the validated spectrometers that have been looked at so far are prohibitively expensive for most small breeding programs to afford. So because of this, we're looking into the use of a portable handheld spectrometer called the SIO, which at $300 is a 150 to 200 times price reduction from the 50,000 plus that these other spectrometers cost. So it does measure a restricted amount of wavelengths in the near-infrared spectrum, area of the electromagnetic spectrum. But as I'll talk about, we've found this to also be highly predictive of dry matter content. And then the spectrometer connects with Bluetooth to a smartphone app that can then be used by breeders and technicians in the field to collect data. So now I'm going to take a little step back and talk about what near-infrared spectroscopy is. So this is a method that allows for quick and non-destructive analyses. It's been used in lots of different fields, including you can tell medications apart, like counterfeit or non-counterfeit. And then in agriculture specifically, it's been used for cultivar identification for measuring Brix and other traits. And what it is the spectrometer measures the reflectance of light in the near-infrared region of the electromagnetic spectrum, which goes from about 700 to 2,500 nanometers. And the reflectance in this region represents a complex signal from the physical and chemical properties of whatever is being scanned. So because the signal is complex, it requires multivariate statistical techniques to be able to decipher what's going on. And then it also requires the development of calibration models for use in each and every application. So this cultivar identification in beans will require a different model from measuring bricks in apples. And so in my case, this looks like 3 main steps. First, the calibration data is collected, and then that data is fed as training data into a prediction model development pipeline. And once those models are developed, they can be used for routine analysis in breeding programs. So this requires protocol optimization to decide what different steps need to be put into the data collection protocol and also into the model development pipeline. And then once the models are developed, they will also require consistent updating as the phenotype change, as environments change. So I'm going to zoom in and talk about my work in steps 1, 2, and the protocol optimization. So I have 2 different breeding programs that I'm working with, IITA in Nigeria and NACRI in Uganda, each with 2 different years of data. So far, I just have this first year collected. My analyses for the rest of the presentation will focus on these trials in Nigeria, which include 129 genotypes total with 2 reps each. These data were collected with 6 roots per plot. The roots were sliced in half crossways and then scanned cross-sectionally. These are more slices than were actually taken. It was just cut in half, but scanned like this. And then those same roots were put in through the standard operating protocol for the oven method to determine dry matter content. And this was done for each individual root. So for each root, I have a pairing of scans and a dry matter content value from the oven method. These were then fed into pre-processing techniques and then into my model development pipeline, which consisted of 50 iterations of random split of 70% training and 30% validation to get a better idea of what different training sets what the effect of different training sets will have on the predictive ability of the models. And so then I looked specifically at the different pre-processing techniques and their effect on the models themselves. So I looked at 12 different techniques, including scattered correction and derivatives, and compared them with the raw data values. And so this graph shows from left to right on the x-axis different pre-processing techniques with raw data on the far left. The top is predictive ability of the model with R-squared prediction, and the bottom is root mean squared error of prediction. And as you can see, there's no statistically significant difference between the different pre-processing methods and the raw data itself. So I will use this to inform my model development pipeline moving forward, but I'll keep checking it with different data sets as the data itself might require different pre-processing methods. So one other thing that I looked at in the protocol optimization is the number of roots that I included per plot. So if you remember, I had the dry matter content for each individual root, and I used that in this analysis. So from left to right, we have 1 through 6 roots per plot, and then this is the R-squared prediction again, and the root mean squared error of prediction. And there are clear advantages to adding more roots, but there seem to be diminishing returns after about 3 roots per plot. And then this, again, will be used to inform the future calibration data collection and the SOPs that we develop for breeding programs to use in their routine phenotyping of dry matter content. And so with this, our final goal is that routine use of this method in breeding programs, but to get there, we have additional protocol optimization that I'd like to perform, and then the additional data from the other breeding program in Uganda, as well as from 2019. And then I'd like to evaluate prediction models across environments and breeding programs. And then finally integrate this data type and the models that I create into cassava base so that breeders can access them directly and use them in their breeding programs. With that, I'd like to thank my lab and my great collaborators from the Next Gen Cassava Project, the PhenoApps team, and my committee and funding.
Are there any questions for Jenna?
Yeah, Jen, how repeatable are these? Did you try measuring the same roots 2 or 3 or 4 times?
So I scanned each route 6 times, and so far have been averaging the scans across a route. But I do want to look at the number of scans per sample to see if that's affecting the results that I'm getting. So looking at 1 scan per route and comparing it with another scan per the route, or averaging 2 or 3 to see how many we should be adding in our protocol. So it's not something I've looked at yet, but I do plan to look. Yes, Rebecca.
You just mentioned 6 scans per root, but when you were showing the slices, you said you were really just doing 1 slice per root. Could you just comment on how the size of the root and where exactly you scan it in terms of the, the diameter versus the longitudinal position? Like, how does that, just like the vegetative age of that root, how do those things influence dry matter and scans and?
So the question is whether or where exactly on the route the scans are taking place and how that affects then the results that I'm.
And also just that size and age of that specific
size and age of the route. Okay. So we're taking the route and cutting it in half crossways in the middle, and then taking 3 scans on each half, both on the cut surface. So we have not so far looked at different regions of the root. So we do know that dry matter can vary from the proximal to distal end of the root. And that is something else that we want to look at. So we do have a collaborator at Embrapa in Brazil who's looking at this right now. And I've been talking to him about getting that data and looking at it. But so far, the roots have all been the same age, roughly the same size. And so that also is not something I've looked at yet, but I plan to.
What is the relationship between dry matter and consumer acceptance?
So all that I know about that is secondhand from breeders and from people who eat cassava, a few of them. But they all say that the higher dry matter is better. It just like feels better in your mouth. It cooks better. And so I think that consumers can tell whether a root is high or low dry matter when they're eating it, and then it might mean that they don't buy cassava from that person who sold the low dry matter cassava to them or something like that. But I haven't done any direct consumer studies or anything like that.
Any other questions? All right, let's give Jenna another round of applause.
This has been a production of Cornell University, on the web at cornell.edu.